Oh, dude, it's fun. So Maria and Madison, are you guys both sitting in the same room, but on your different devices? Or are you in different places right now?
She's in the library.
Oh, all right. So you are legit. You're in one of those like
Whatever.
study rooms as the
Mm-hmm.
like the boards that you can write on and everything, huh?
Yeah, because I just got off
Well,
tutoring, so.
got it. All right, so we'll just wait a minute to see if anybody else joins, and if not, it is a private session just for the two of you. It'll be made available for everybody. I'm going to run it the same way I have in the past, as we'll sort of go through sort of what the review study guide and all of that stuff is about, and then we have one. Oh, is that you, Maria?
Huh?
Something super noisy. What do you do? Like there's papers rustling all over the place. Now are you muted? Yes, now you're muted. All right. Um, well, we'll just give a second. I'm going to open a bunch of documents that I will end up sharing, and then we'll get started here. I have two crazy dogs that are insisting on trying to wrestle. I'm in my dining room right now, and they're just being crazy. Here, Daisy, you want to come say hi? In here. In here. Well, here's Charlotte. Charlotte's very big right now. So here's Charlotte. All right, Daisy, you want to come say hi? Come here. You're crazy. Come here. Come here. All she wants to do is roughhouse with Charlotte. Come here. Fine. You don't get a chance to say hi then. There's Daisy. You're crazy and you need a haircut. You need a haircut. Go play. All right. Biochemistry content. Homework.
Dr. Perani,
Yep.
just to let you know, me and Danny today, we tried working with the mutants for the enzyme assay.
Yeah.
None of them
Mhm.
worked.
I'm not surprised the enzyme was not stored in really great storage buffer. Did you really try very, very high concentrations?
Yeah, we tried keep adding like
Did you
200
try
microliters.
about dilute?
Oh no, we didn't.
Okay. Did you try using MG8? Did that work?
No. And we tried like some of the fractions that we had in
No.
the, and it didn't work neither.
Of the MG8? Okay. So the goal is we want to just see if we can get something to work.
Mm-hmm.
So we'll check it out. We'll check it out tomorrow.
Okay, yeah, because me and Danny, well, Danny sent me the picture that she took of the graph that displayed
Got it.
how, like, it decreased, but then it went constant.
Got it. Okay. So it maybe did a little bit, but then it stopped. Okay. So it's very possible that we need to repurify new enzyme, and that's okay. So maybe one of the first things that we need to do as well is make some more of the wild type at the very least, since we know how that behaves. Okay.
Okay.
And I think I haven't looked back here. So yep, it's still just the two of you. So I'm going to share my screen. And then we'll just sort of go through sort of this study guide. I'll kind of, just do what I've done in the past, unless you guys have something different that you'd like to do. So I've got the exam study guide here. I've got the homework in the annotated notes for each of the 4 units. And then there is something new that I have, and I will show it to you guys at the end, and then I will post that in Teams. Just sort of a new study tool, study strategy that I start when we have exam 2, and I do it for exams 2, 3, and 4. So I'll let you know what that is when we get there. But any big picture questions on anything before we get started here with this? Okay.
How different is going to be the exam compared to exam 1?
So it is different, which is why I'm going to give you a study tool that will help you with that. I will highlight that a lot of the questions are going to be things where you maybe have to, and I'll give you an example of one of the questions. So I'll get to that, but it's almost like a single word fill in. So there's lots of questions like that where there might be a 1-word answer to answer a question. And I'd say maybe 60% of the exam is like that. And then the other 60% is, as you'll see here, drawing structures. There is a mechanism, thinking about some of those slides that I told you to put stars on because they were really important, like the Perutz model or things like that. So it's probably a 60-40 mix of those 2 types of questions. I just have to like they are fighting here, like as much as like puppies and a 3-year-old dog can do. Oh dear God. All right. So structure drawing. We did nucleic acids and then we did peptides so far. So know the structure of ATP and relevant numbers. Okay, so this is the only nucleotide that you need to know how to draw. I would expect you to draw any nucleotide if you were just to write the word base that's up there. But I do want you to know the structure of the nucleobase adenine because we see that in so many things. So that's the only one you need to know the structure of the base for. This was a homework question. We'll kind of highlight it in a minute. You are going to have to draw a dinucleotide of DNA or RNA. You're going to see information that tells you, like, is it 5' and or 3' phosphorylated? Is it DNA or RNA? And again, it's going to be just like the homework question you had. So we'll sort of talk about that when we look at the homework. Draw a peptide. It's going to be a fun word that I will think of, somewhere between 4 and 6-letter words. And you have to draw that peptide indicating relevant features like directionality, labeling the N and the C termini, labeling a peptide bond. You get full credit when you can draw in what that amino acid structure is. Partial credit if you just tell me the functional group that's there, and then like almost no credit if all you do is like put the letter for the amino acid in. Okay, so you still need to know your amino acid structures. And then when we think about structures, be able to think about the similarities and differences between secondary structural elements, such as alpha helices and beta sheets. Okay, so as we go over sort of the homework and the notes, I'll sort of draw attention back to these, but definitely are some structure drawing pieces that you'll see there. Any questions on that right now before I move on to the next section? I think this is only a single page thing here. So there's structure drawing, structure concepts, function, and then application. So that's sort of the format in terms of compartmentalizing the things that we'll see sort of for the exams moving on. So there's structures that you may have to draw. structural concepts that you need to think about, knowing that structure affords function. There's function that you need to think about, and then thinking about some applications. And I will tell you right now, as I go through this study guide, I'm going to tell you that there's things that you can cross off. I haven't changed what this document is from year to year, but I know that there's things that I'm going to take off your radar because I know I didn't cover them. So we'll address those as we get there. Concepts, discuss what is meant by the limits of sequence space. So all that has to do with is when we played those numbers games and we said, how many different possibilities would there be for 100 mers of DNA or for a protein? And just conceptually understanding that we don't even have enough atoms in the universe to test every single possibility. So I want you to know that concept, but then also sort of be able to do the math that I said, hey, if you wanted to make something that was, 15 amino acids in size, and you were going, do the math. It's an extra factorial thing, be able to think about how to number that. Go in those. Note that says discuss the concepts of sterics and energetics as they rate structure formation. Okay, remember the concept is if we want to have a stable structure, we need to make sure things aren't banging into one another. Specifically for, what are you laughing at? I appreciate that. I can see your face, but I can tell you're laughing.
You keep cutting out, so we missed the whole thing you said about the limits of the sequence space and stuff.
Okay. All right. Well, you need to tell me that and not just laugh, Maria, because I don't know what's going on. Okay. So it was saying that my internet was unstable. So I need to find out if David's on video games. And if he is, he needs to get off so I can do this. So hang on one second. David, are you on a video game right now? Are you on a video game right now? Oh, okay. Oh, all right. He says he turned off his internet. Okay. Sorry. So limits of sequence space. Can you hear me now? Yeah. Okay. So conceptually, I just want you to know that we don't have enough atoms in the universe to check all the possibilities. Conceptually, that's all you need to know. I do want you to be able to know how to do a little bit of the math. So what is your deal? You look like you're crying. Am I still frozen? Am I garbly? Now I can't hear anybody. Are you
you're
muted,
okay.
Madison?
I'm not.
I'm all good? Okay.
Yeah.
Is it still bad for
No, I can hear you, I think, right now. Your
Okay.
camera's freezing, but it's fine.
My camera? And is it freezing in like a really bad like face? Is that what's so funny? Oh, good gravy. All right. Let me try to plug in my phone here, and then maybe I will sort of try to connect to my cell phone. Sometimes internet isn't great, or maybe I'll try to go upstairs. Maybe I'll just do that because I think the Wi-Fi connection is better upstairs. Great. Now people are going to watch this afterwards. I'm going to be frozen in some crazy face. All right, so let's go upstairs here. Hopefully it gets better. I should know that the dining room is not great. Hang on. Hey, I need you to pay attention to the dogs right now because I have to go into the sitting room here. Well, then put Daisy in her crate.
Okay, Daisy, you guys want crate? Come on.
All right. Hopefully this will be better. All right, so let me know again, and not by just starting to laugh. Let me know, like, give me a thumbs down if because you're not frozen to me. So, but anyway, I want you to be able to think about some of the those calculations, whether it's using factorial or using exponents. Like if you had to tell me how many possibilities are available here, so be able to do that math. Then we were talking about concepts of sterics and energetics. So anytime we're going to form a structure, we need to make sure that we are not having atoms crash into one another. That's sterically unfavorable. And then from energetics, we need to think about the enthalpy and entropy pieces. It's always unfavorable to form structure from an entropy standpoint. And so we compensate with enthalpy. And I always want you to be thinking about enthalpy as intermolecular forces and entropy as more molecules moving more. So if we don't allow for molecular motion, that's unfavorable entropy. And then from an enthalpy standpoint, we compensate by forming those four intermolecular forces. And Maria, you probably heard me going through them today with a Gen Chem student. They still have to know those same four: London dispersion, dipole-dipole, hydrogen bonding, and then ionic interactions. Think about the hierarchy of protein structure, right? So primary, secondary, tertiary, quaternary. When we think about these four, primary structure is really just sequence. It's not even technically a structure. It's held together by a covalent bond. You start having three-dimensional structure with all three of the other ones. Secondary is, again, backbone atoms that are hydrogen bonded together. Tertiary is going to be all of the intermolecular forces and side chains interacting. And then quaternary structure requires that you have a multi-subunit protein. So again, we'll look at some pictures here that are going to allow us to remember and reinforce that. And I'll make sure to highlight and call back to these kind of study tips to make sure that you kind of know that those are the things you need to study. We address this with the kinds of intermolecular forces used to create tertiary structure, address their effect on stability versus specificity. Maybe I'll bounce back and forth here with some slides, right? So particularly that is, so this was the slide that was really important for the hierarchy of protein structure. So even if you want, you can write down slide numbers that are important for that. So that was slide number 8. Here was the idea of enthalpy entropy compensation with energetics, and then sterics specifically for proteins. We talk about the Ramachandran plot as giving us sort of allowed angles that prevent unfavorable steric conformations. So this slide here, let me come back. I think we're talking here about stability versus specificity. So that was this slide, right? So this slide reminds us that stability is largely driven by the hydrophobic effect in tertiary structure. Stability is hydrogen bonding when you have secondary structures, but specificity, who's next to who, actually some of the strongest intermolecular forces, especially things like covalent bonds with disulfide links, they don't contribute to stability of proteins. They're largely contributing to specificity of, okay, who is going to be next to who? And we spent a lot of time talking about that with the insulin case study. Let's see. So we just sort of talked about define the hydrophobic effect and then what its role is in protein structure formation, right? Tendency of nonpolar substances to minimize their contact with water. So if we're talking about those nonpolar substances being nonpolar amino acids, that's going to contribute to protein structure formation because all of those nonpolar side chains are going to want to come together in the middle of a protein. Okay, and that's what we call hydrophobic collapse. And that really starts that globular protein formation. Fibrous and globular proteins, we talked about some examples of each. Collagen and keratin are fibrous proteins. Globular proteins, hemoglobin and myoglobin are two that we've sort of talked about. And just like their name implies, like fibrous proteins are going to tend to be alpha helices. They're going to be very long, repeating sort of amino acid residues. And then globular proteins, like the name implies, they have sort of a fixed number of amino acids and they have a very compact shape. We had a couple of slides that compared and contrast how structures are determined by X-ray crystallography versus NMR spectroscopy, be able to sort of talk about the pros and cons of each. And think about this concept of structural resolution. Anytime you have a protein crystal structure or a protein NMR structure, it'll tell you that it is solved to a certain resolution. It's going to be a number. It's going to be a number in angstroms. The smaller the number is, the more you know for certain where atoms are. So the higher the resolution, right? You can think about this in terms of a picture. A picture that has very high resolution is not fuzzy at all, right? Everything is well defined. And the same is true with protein structure. When we talked about hemoglobin, we had terms apoprotein, that's the protein without its ligand. Holoprotein is the protein with its ligand. And then we talked about those ligand sandwiches. So specifically for our globin proteins, we have oxygen binding to iron, iron binding to heme, heme binding to the protein, and that's sort of the ligand sandwich that we have. And then lastly, and very recently, we sort of talked about T state and R state. Those are going to be concepts we will see throughout the entirety of this whole course here, this year-long course. But we learned it in the context of hemoglobin. So T state we can define as a conformation that is not conducive to binding substrate. R state is conducive to binding substrate. Okay. So those are some concepts that deal with structure. I'll see if you guys have any questions here, and then we'll sort of revisit these as I sort of go through homework questions and highlight either ones that are really important to know, or we'll highlight slides that are important. But I'll pause there, see if you guys have any questions.
Do you think we could go back later to the slide where we talked about the limits of sequence space?
Yep, 100%. I'm going to go through each of the slides, and because I didn't have that slide up, I'll remind you that, hey, this slide is related to this sort of study guide topic, and here's some of the things you need to know how to do. You're not going to get a word scramble on your test, okay? But I might ask you how many possible sequences would be available to make something that was 7 amino acids long and used all 20 combinations, right? So we might need to think about that. She just wants to be here. That's fine. Is Daisy in her crate? Okay. Okay. We have started learning about our B vitamins. So I'll highlight for you guys that the sooner you learn about these, the easier your next fundamentals quiz is going to be, which will be on vitamins. So you are going to need to know vitamins. We've learned 3 B vitamins in the nucleotide learning unit. We learned vitamin B2, which is found in FAD, vitamin B3, which is found in NADH, or NAD plus or NADH, and then vitamin B5, which is found in acetyl-CoA. So we'll kind of highlight those when I look back through those slides. You don't need to know structures of anything there, but you just need to know how to match the pieces. And then again, know what those different molecules do. So FAD is really a redox carrier. It carries electrons. So it's like an electron middleman, carries 1 electron at a time. And then vitamin B3 is for NAD. It carries electrons 2 at a time. Hang on, I'm going to call David and I'm going to tell him that he can let Daisy up here. Maybe she'll stop barking. Minimum. Hey, if you want, let Daisy out of her crate. You can bring her up into my room here. Okay, bye. Minimum and maximum sizes for proteins and understand why. Again, we'll see this on that slide, Maria. You need at least 40 residues to reasonably be able to have those intermolecular forces that will allow for structure. And then more than 4,000, and you tend to introduce mistakes that might not allow you to have a functional protein. So that's what those numbers are. Discuss relevant aspects of proteins that we learned about. The two filamentous proteins are keratin and collagen. Since I know those slides are kind of handy here, let's pull those up. So for both of these, oh my goodness, you need to lay down. You need to lay down. So the key here, and I do want you to know the specifics for each one. So keratin is that coiled coil structure. So there's two alpha helices that are wound together. Both of them have this right-left sort of combination, but you need to know which is which. So alpha keratin, and the way that I sort of remember it every year when we go through this is it's like, okay, there's only two helices there. So it's easier to have the right-handed coils. So we're going to have two right-handed coils. And since I always have the opposite handedness, that means I've got two right-handed alpha helices that I wrap in a left-handed coil. Okay, so I want you to know those specific pieces. I want you to know the specific amino acids that we tend to see in them. The important amino acids that we're going to see in alpha keratin are serine. I'm sorry, not serine. We have disulfide bonds. I'm looking at that S there. We have cysteine residues. Okay, so that's an important thing that we find in keratin. We also see at the first and the fourth positions, we're going to have nonpolar residues. We didn't specifically talk about what they were, but we see at that first and fourth position nonpolar residues so that we create a region of hydrophobicity that will allow those nonpolar sections that are on the inside of these alpha helices to find each other and help form that association. So that's what we see for alpha keratin. For collagen, collagen is the one that has three. Because it has three, I remember that it's a triple helix. It's going to be right-handed. And because I have three helices, I'm going to have the more tightly wound one, which is the left-handed alpha helices. So three left-handed alpha helices and a right-handed triple helix. This one, we did have a very specific set of residues that I want you to remember that it's glycine, it's proline, and hydroxyproline and hydroxylysine. So we essentially have a small residue, proline, which adopts the cis geometry, which is going to be really important when we form that left-handed alpha helix. And then we're going to have hydroxyproline and hydroxylysine. I don't actually think we talked about this. So it's not specific that I don't even think there's any questions on it. But do you guys know what the disease is? Well, I'm going to tell you about this and I'll remind people in class because I probably, I don't think I had it on the next slide, probably should have talked about this. I have to remember whether I wrote this in as a question or not. Do you guys know what the disease is that's caused when you have faulty functioning collagen?
Is it collagen deficiency?
So well, and I should say this is collagen deficiency diseases, and there's lots of those. But I should say vitamin C is required to make functional collagen. Let me ask the question a different way. Do you know the disease that's caused from vitamin C deficiency? Scurvy. Have you heard of scurvy? Okay, so scurvy is from vitamin D deficient or vitamin C deficiency. Scurvy, when you have faulty collagen, the first place you notice with faulty collagen formation is your skin and specifically your gums. So people will notice that they have bleeding gums a lot. That's one of the first signs of scurvy, and it's because you have faulty collagen, you have faulty connective tissue in your skin, and it causes bleeding. People don't die from scurvy. They will die from infections that happen because you don't have a nice intact barrier to the outside world. So I don't think we told that story. That's why sailors early on, they used to take citrus fruits with them, specifically limes, because they would last for a long time on a trip. They didn't know why, but they would suck on limes and they tended not to get this awful disease. And that's because citrus fruits are very rich in vitamin C. So little story there. I'll make sure to remind people that there was a story that I talk about with collagen that you want to make sure to watch the video to listen to in case there's a question or two on that. So that was collagen and keratin. Let's see. I'm going to wait till we get to the sort of this slide to sort of talk about this. But the 5 equations that take into account breathing, pH, oxygen delivery, CO2 waste elimination via carbonic anhydrase activity, and then ATP use and consumption. That's going to be an important equation for you to address and think about when you do your Flipgrid video. That making that video is going to be a good opportunity for you to study this content. I skipped over this one, not intentionally, but just seeing it here. Difference between oxidation and oxygenation. So again, we'll go back to that slide. We had some graphical information here talking about KD, fractional saturation, PO2, and P50. So there are some numerical values I'm going to want you to know. Think about what the ranges are that things run between. So we'll look at that slide again and highlight those pieces. Equations that we have, both chemical equations to think about things. And so I'll highlight what these are when we look at that slide. Think about what these pieces mean mathematically. So KD, PO2, P50. So I've got a couple of slides in particular that we'll look at to highlight what those pieces are. Again, 2 graphs that we'll look at to indicate the difference between cooperative and non-cooperative binding. Those are particular for myoglobin and hemoglobin function. We've talked a lot about what happens when we shift curves. What does it mean when we shift a curve to the right, shift a curve to the left? Big picture, when we shift a curve to the right, that means we're binding our ligand less tightly. We're binding it more weakly. If you shift a curve to the left. So just looking at those curves that we have, we can tell that myoglobin binds oxygen more tightly because it's a more left shifted curve. So we'll look at those graphs again and we'll sort of talk about these. Just yesterday in class, we talked about concepts of carbon dioxide, BPG levels, pH, oxygen concentration, all ways that modulate oxygen binding. So we'll sort of take a look at those in a little bit. We didn't talk too much about the Human Genome Project, but these are some numbers that I want you to know. So even though we kind of glossed over this slide a little bit, I'm going to tell you the story now, and I want you to know these numbers. So the Human Genome Project is a project that took 13 years and $3 billion to try to sequence the human genome. And we knew that there was 3 billion base pairs that were there, but again, we didn't know what the sequence was. After doing all of that work, again, 13 years, $3 billion. You guys know what it costs to sequence your genome right now? You could have your entire genome sequenced. Do you know about what it would cost if you had a guess?
It's not that much.
Is
I
it
just
No,
can't
it's-
remember.
What's that, Maria?
Is it $5?
Oh my gosh, Maria. $5. That's what it would cost us to send our plasmids off for sequencing.
Yeah.
Right?
Oh, yeah.
Now, you can send like 23andMe. That might be like $129 or something, but that's not your whole genome. It's about $1,000 to do your entire genome. So you ruin the punchline, Maria, by saying $5. It's not $5. And I think Madison was like, it's not a nominal amount, but it's certainly not like
Yeah.
unreasonable for like somebody to say, if I really wanted to know what was going on, I could sequence my genome. But here's the thing that's kind of interesting. At the end of the day, once we had all that information, there weren't as many genes there as we thought, right? From a genetic standpoint, all you guys have to take genetics at some point. I don't know if you've taken it yet, right? But there's open reading frames. That's how we can tell that we're going to a place where there's a gene, and then you can find where stop codons are. Of all 3 billion pieces of information, only 1% of it actually codes for useful proteins. So it's kind of interesting. Sorry, I thought the dog was getting into something. It's really interesting to think that there's so little information that's there. What's really interesting too is about 8% of our genome is ancient viruses that over evolutionary time have integrated in our genome and then persist. So it's not even like self DNA. Oh, no, you cannot have that blanket. You cannot have this blanket. No. Nope. So didn't spend a lot of time on that, but it is something I do want you guys to know those pieces of information. So if you know the stuff that's on your study guide here, you'll be all good. Okay, to be able to define SNP, single nucleotide polymorphisms. So know what that is. Okay. Discuss how prevalent they are. I think there's a slide we'll show what it is. It's about 1 in every 300 nucleotides. It's kind of different between people. And so you're being really bad. Am I going to have to get David and get you back in your crate? Because you're not behaving yourself. No, you're not. You're not. Lay down. We had 2 stories that we talked about. So I kind of do want you to know some aspects of those 2 stories. I want you to know a little bit about MTHFR. So we'll look at those slides. We'll go over that again. And then ApoE. So we'll use, when I look at those slides, as an opportunity to remind you of the stuff that you need to know. Oh, you know what? And don't worry about E. I'm going to tell you this, but this is not anything we really went over. We talked about the concepts of epigenetics. Epigenetics is the ability of the environment to control whether genes are turned on or off, right? So there's a quote that is used a lot in sort of the epigenetic space that talks about genetics is the gun, but it's the environment that really pulls the trigger. And I think I've stopped talking about this because I don't like references to guns anyway. I don't think it's useful, but it's an interesting analogy because the same way you may have the gene, but unless that gene is transcribed into a message, translated into a protein, is actually an active protein, you know, that whole going through all of those omics, it doesn't matter that you have the gene. So just something to think about there. And then just yesterday, we talked about sickle cell anemia. Okay. So I'm going to take a second here and we'll go through all of these slides. We'll hit back on some of these pieces. Why do you keep bringing in clothes? Like she's just grabbing things off of like a pile. No, this is not for you. You're a bad girl. All right, let's look at new, and we'll take this one learning unit at a time. And then if you have questions, we'll address them before we go on. So I'm going to flip through slides here. And then we'll sort of look at the homework, and then we'll see if you have any questions that are based on stuff. All right, so highlighting, you do need to know how to draw a structure. You're going to see something where I'm going to tell you how to draw it. Remember, you're going to look at the front and the back end and see whether there's a P there. That'll tell you whether there's a 5' or a 3' phosphate that you need. You're going to look to see whether there's a little D there. If there's a D there, that means it's DNA that you're drawing, and you need to have a hydrogen in this position. Other than that, 5' to 3' is the direction. This is a phosphodiester bond. Whatever the letters are in the dinucleotide you have to draw, you have to make sure you put those letters in the right position. So hopefully pretty straightforward. The backbone is pretty much what we're drawing here. Understanding that nucleosides are bases and sugars, nucleotides are base sugars plus between 1 and 3 phosphates. So pretty basic structural information there. Again, structure of ATP. You should already know like the phosphate connections. It's a ribose sugar. And then this is the only other piece that I want you to know is the structure of that nucleobase portion. You do need to know that magic number of minus 30.5. I do also want you to know this enzyme called pyrophosphatase. This is kind of a ubiquitous or found everywhere enzyme that if you have processes that generate pyrophosphate because you have this enzyme around, it immediately removes that from solution, and then it allows reactions to get pulled forward. So we're going to see pyrophosphate and pyrophosphatase a lot throughout the semester. And it's just a way that we impart irreversibility to chemical processes. The other thing is that these are the high energy bonds. These are the phosphate ester or diphosphate ester bonds. And they are valuable because when we break them, we do charge separation. That makes something lower in energy, which is why we get energy out from it. This is the piece that I was just sort of talking about. These are 2 important concepts that we're going to see throughout the course, which is why I spent time sort of talking about them. So just highlighting, you don't need to know this aspect of discuss the quote, blah, 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 blah. Okay. What I do want you to know is this sort of omics flow. So DNA to RNA to protein to metabolites, genomics, transcriptomics, proteomics, and metabolomics, right? And this is the easiest information to know. It's really easy to sequence somebody's genes, but that means nothing unless you go through these other processes and ultimately have those metabolites that are what are directing biochemistry. So that's sort of the big key picture with this part. And then this is something we are going to see throughout this entire year. So I introduce it early, and we're going to continue to see this. When we talk about how we can control whether these enzymes are active, very often they're controlled by something called covalent modification. That's the addition or the removal of phosphates, using kinases to add phosphates or phosphatases to remove the phosphates. Those phosphates are added or removed from three important amino acids that have hydroxyl groups: serine, threonine, and tyrosine. If we're adding phosphates with kinases, we are in a phosphosystem. If we're removing phosphates with phosphatases, we're in a dephosphosystem. It's really important to be able to sort of understand this overarching umbrella, because when we are in a need state, we do a lot of kinase activity to put ourselves in a phosphosystem. Why understanding this umbrella is important is because once we know what our body needs to do, need state, fed state, it's going to dictate whether we're decorating things with phosphates or removing those phosphates. And that's going to help us understand what we need to do to turn on or off certain enzymes. So this sort of becomes a knowledge cascade, if you will, to sort of think about these pieces. Okay, I'm going to pause there for a second because I've had a couple of busy slides. See if there's anything you guys want me to go over again. Good?
Can you go over drawing the dinucleotide?
Yep. So again, you're going to see something. We always write, whether it's proteins or nucleic acids, we always write them in the same direction. For nucleic acids, it's 5' to 3'. So when you see something, I'll say 5', and then you'll see a combination of letters and stuff after it. First thing you want to look at is whether there's a D in the beginning. That's going to tell you whether you have DNA or RNA. Whether you have DNA or RNA, the only thing that changes is whether you have a hydrogen or a hydroxide group here. With me? Oh. Next thing that you're going to look at is there's going to be stop it. There's going to be a bunch of P's that'll be present. Always be a P between the two letters. That represents the phosphorus of the phosphodiester bond. Okay. Then you have to look and see whether you have a phosphate at the start, which will tell you that you have a phosphate at the 5' position. If you have a P at the end, that's going to tell you that you have a phosphate on the 3' position. Does that help? And then the other thing is you sort of need to know how to draw this. It's an OH group and then a CH2, and then we're going to have a 5-membered ring. Okay, that's a ribose ring, oxygen here. And then we're going to have an oxygen with our phosphate here, and then we repeat. Okay, this is your 3' hydroxyl. This is your 5' hydroxyl. You need to go downstairs and see your father. I'm not dealing with you right now. Shh. Go see daddy. All right. Does that help, Madison? Okay. All right. Next few slides are going to go quickly because these are the ones that just have to do with your B vitamins. So I want you to know that vitamin B2 is associated with FADH. Vitamin B3 is NADH. I should have said FADH2. Know the oxidized and the reduced forms of each. So FAD, FADH2, NAD+, NADH. This redox activity for the vitamin B3 derivative happens 2 electrons at a time with hydride. It's 1 electron at a time with radicals for the FAD, FADH2. That's all you need to know for that. You don't need to know anything about structures. By the time we do get to our vitamins quiz, you will want to know that vitamin B2 is riboflavin. And then we've got niacin or nicotinic acid for vitamin B3, but you don't need to know those now. And then we've got acetyl-CoA is vitamin B5. You don't need to know this structure, but highlighting that we have a thiol group that's at the end of this really important biological molecule, and that's how we're going to attach on an acetyl group and generate acetyl-CoA. And if you've taken any sort of intro sort of cell and molecular biology class, you've probably talked about acetyl-CoA before. So that's those guys. This was sort of a big picture thing that talked about, we've talked about that fuel equation. Fuel plus oxygen gives carbon dioxide, water, and energy. So what I want you to think about when we do fuel metabolism is our fuel is like a sponge that's held together with electrons that are water in the sponge. What we're going to do when we do fuel oxidation is I'm going to squeeze the water out of the sponge, but I want to collect that water. I want to collect that electron. So we collect those electrons by forming redox cofactors. So again, our vitamin B3 and our vitamin B2 molecules here. So we collect electrons by reducing and generating NADH and FADH2. The carbon atoms that are now more oxidized go on to acetyl-CoA. The citric acid cycle, which we'll learn about next semester in detail, converts all of those carbon atoms to single carbon atom molecules, carbon dioxide. In that process, we'll also squeeze out some more water and we'll generate even more NADH and FADH2. These guys go to the electron transport chain. The purpose of the electron transport chain is to generate a proton gradient. That proton gradient is used to generate energy. So we are starting to take this initial equation that we learned about and understand how it works. I will tell you by the end of this course here, you are going to have a concept map slide that has this fuel equation in the middle and tons of arrows moving around and about to really understand how we take fuel and make carbon dioxide and how we generate energy. But this is sort of the extent of it right now. Any questions on this part? Okay. And again, here was the Human Genome Project and epigenetics piece. So again, you can see some of the details for things that 20,000 genes, 3 billion pieces of information, the idea of, only about 20% of genes are what we call deterministic. We studied sickle cell anemia. And so that mutation is deterministic. You will have sickle cell anemia or at least be a carrier for it if you have that mutation. There's no environmental sort of influence on that. SNPs, again, be able to define what an SNP is. The frequency, again, is 1 in every 300 nucleotides are different. So lots of genetic variation there. And then we had 2 slides. These are, I think they're the last 2 slides here, talking about MTHFR and talking about ApoE. So I know that there's videos and stories on both of these. The big picture is connecting MTHFR to the synthesis of a biological methylating agent. And then we had, and we will touch on each of these stories later on in the semester or next semester in our course, but talking about how methylation agents are sort of used. And then the big one that we sort of talked about from a disease standpoint is people who are MTHFR deficient tend to have increased risk for cardiovascular disease. And that has to do with the increased levels of homocysteine that they might have. So that was MTHFR. The big connection with ApoE is, again, understanding the correlation and connection with Alzheimer's disease. So I'm going to pause there for a minute, see if you guys have any questions on any of those slides that you want me to go back over. Remember, these slide decks should be available for you through Teams, so you should be able to find stuff. Let me just take a quick glance through homework. Again, highlighting structure of ATP, Madison, to your point, here's an example of drawing a trinucleotide. I will tell you it's only going to be a dinucleotide that you'll have to draw. Just takes less time, doesn't take as much space. Again, noting that you will see something in this format, so you need to know how to pick apart that sort of code, see what's going on. These were actually questions that were related to things that I know you saw on your quiz. So we can more easily separate DNA strands at a high pH because we're deprotonating ionizable groups, and that affects their ability to hydrogen bond. And so we can separate those strands, and we can degrade RNA because we sort of have this internal nucleophile that then can become deprotonated and more nucleophilic. So those are sort of the reasons for those. This is again just a summary table that allows you to put that information together about B vitamins. Here again is our kinase and phosphatase questions. So we didn't spend a lot of time. I think it was just a 2-day learning unit, but I'll see if you guys have any questions, anything you want me to look at or go over again. Good.
I was just gonna ask if we would get the opportunity to see the questions from the quiz.
Oh, that's good. Yeah, let me see if there's a way. I think in Blackboard, I just need to put a setting so that you can look and see what questions you missed and see what the right answers would be. Okay. If you can put on a little note for yourself, just so I don't forget, send me an email, because then that becomes like a to-do list item for me. So just start an email, and then if there's anything else we come up with here, you can send it at the end and I'll address those. All right, so that's nucleic acids. Let's take a look at protein structure. I'm going to run through the notes if there's slides that we haven't talked about already. This was a big slide because here we sort of ran through at the very bottom of this slide how to draw a peptide. You are going to have to draw me a little peptide. It's going to have some like cute name, like here it was rain, right? But we do NC-alpha carbonyl. So draw your backbone first, labeling your N terminus and your C terminus, identifying where a peptide bond would be. Again, this structure, and because we hadn't really talked about amino acid structures yet, I didn't bother drawing them in. But this would actually get no credit for the side chain part of drawing this structure, because all that you did was put the letters in the right spot. Partial credit is you'd have to say, oh, R is arginine. That's that guanidino group. Oh, A is alanine. That's a nonpolar. I is isoleucine, nonpolar. And then I've got asparagine. Oh, okay, that's going to be an amide functional group. Remembering the functional groups will get you half credit. Full credit means you need to actually draw in the structures of the amino acids. Okay. Some other pieces to sort of know. The alpha carbon is chiral. Stereochemistry for all amino acids is L. I do want you to remember that. We're going to talk about carbohydrate structure tomorrow in class. Obviously not part of your exam, but that is going to be a difference, as we will see the D configuration. I think everything else is sort of pretty straightforward in terms of maybe things that we've already talked about, in terms of you should know what the pKa values are likely to be. And then thinking about some of the chemistry that can happen with side chains. When we think about cysteine side chains, we can have disulfide bond formation. So that's redox chemistry to go between a thiol and a disulfide. And then the important molecule that we talk about there is glutathione. And so that is your body's natural antioxidant. We're going to see that a few more times throughout the semester, so introducing it here. How it works is we're going to take 2 glutathione molecules. We've got a structure of them in just a couple of slides here. 2 glutathione residues. We oxidize them to make a disulfide. And by doing that oxidation, whatever was the damaging oxidizing species gets reduced. So that's how that works. And I actually heard Dr. Jones talking about this, talking about a PI for proteins, right? Or actually, it's, a PI value can be for any polyprotic substance, right? Anytime you have something that has multiple ionizable groups on it, when the sum of the pluses and minuses on those ionizable groups equals 0, your molecule has no net charge. And that's an important property for a protein. So Pi is a really important property for a protein. But I mean, proteins are really polyprotic acids. So a single amino acid, excluding something that has an ionizable side chain, is a diprotic acid. And then once you start stringing a whole bunch of them together, and how many of them, I'm going to ask you guys, show me with your hands, how many amino acids have ionizable side chains? What do you think, Madison? It's only 5, right? Aspartic acid, glutamic acid, those are our acidic amino acids. And then we've got 3 basic amino acids. We've got lysine, arginine, and histidine. What other one were you thinking of?
I confused glutamine.
Okay. Okay. But then you'd have to put asparagine in that same category. But we're going to have 5. This is more just specifically looking at that peptide chemistry. So this is looking at the peptide chemistry with glutathione. So here's the structure of glutathione. Glutathione is actually a little tripeptide. So it's a glutamic acid, it's a cysteine, and it's a glycine. Okay. Interestingly enough, we form this bond, that amide linkage, not with the normal carbonyl of the glutamic acid residue. We actually form it with the side chain, which is a little bit unusual. We spent a little bit of time talking about how we make the chromophore in green fluorescent protein. Conceptually, we talked about red shifting. That's a way to increase the conjugation so that we can make something move into the visible spectrum. So that's essentially what's happening here, because normally nucleic acids absorb at 280. Nucleic acids absorb at 260. But when we extend this conjugation system here, we get a chromophore that we can see in the visible spectrum because it turns green. All right. Maria, here was, hi. I know, she pulled it down. Sorry, Sam stopped home. I have a class right now.
It's all wet and stuff and half of something, so.
Oh, Daisy, you ate the button off of the hamper. Go just take her.
Half the button. I don't know where the other half
It's
is.
probably somewhere. I don't think she ate it, but I need to finish this class. So take her. Are you here for a little bit?
No.
What are you doing?
Starting a little laundry and then going to watch a movie.
Okay, careful because Leo now lives in the laundry room. Okay. Take Daisy, please. All right. So here was this slide that you were asking some questions about. Upper and lower limits and why. The different kinds of math to think about is whether it's a factorial. And the difference is when you're doing factorial, that means you cannot repeat something. Okay. When you are allowed to repeat, that's when you can use the exponent. And let's see. So this really is, you had asked the question, can we explain a little bit more about nature? How did I word it? Limits of sequence space. That's really what this slide is kind of about, is we don't have enough atoms in the universe to check every option. So nature doesn't. It doesn't have to. It cannot. And so it gets to something that's just good enough. Does that help, Maria? Anything else on this slide? All right, this is the hierarchy of protein structure, right? So I think I sort of talked about that when we were on that study guide slide. So big picture, I want you to know that secondary, tertiary, quaternary are all three-dimensional. What differentiates them is what is interacting and forming intermolecular forces. With secondary structure, it's backbone atoms. With tertiary structure, it's side chains within a protein. With quaternary structure, it's side chains between proteins. Okay, so being able to think about those differences. And then primary structure is a little bit confusing even to talk about because it's not even specifically a structure. It's one-dimensional. It's just saying what is the sequence of the amino acids. We talked about Ramachandran plots, right? A Ramachandran plot is basically a way to show the allowed angles that atoms aren't banging into one another. And then as long as we want to favorably form structure, we have to have overall favorable energetics. And entropy and enthalpy are the two contributing factors to overall energy, into free energy. And so it's unfavorable entropy, so we must compensate with intermolecular forces. And again, I'm going to try to go through these slides quickly because we're really up against the end of time. I want to respect your guys' time. So if there's a slide here that you want me to go back and say something more on, let me know. We highlighted secondary structural elements. Be able to compare and contrast. Secondary structures are all hydrogen bonding. Alpha helices are within the structure, and it's backbone atoms. So again, this represents things that are the same. It's the backbone atom. So it's hydrogen bonding between the nitrogen's hydrogen as the donor, the oxygen of the carbonyl as the acceptor. But we have intrastrand hydrogen bonding for an alpha helix. We have interstrand hydrogen bonding for beta sheets formed by beta strands interacting. But both of them, again, are going to have repeating allowed angles. That's the Ramachandran plot island that we're allowed to live on. And then we compensate with hydrogen bonding. Side chain distribution for alpha helices, it's out and down. For beta strands that interact to form beta helices, it's alternating on the side of the sheet. By and large, we're going to put hydrophilic things on the outside, non-polar hydrophobic things on the inside. That's all we think about here. We already went through these two slides on collagen and keratin. Collagen and keratin. Conceptually talking about how, hey, you know what? Nature figured this out millions of years ago when we made rope and we found the strongest rope. We actually did the same thing. Comparing and contrasting X-ray crystallography and NMR spectroscopy. I want you to understand how each works. So this is what you need, what you do, what happens, and any limitations. And then this is a good slide to sort of compare and contrast. This is another slide just showing you the hierarchy. We went through this slide talking about the differences between stability and specificity. Really didn't get to this. This was that end of the day that Dr. Brown was there, and I really wanted to show him this slide because this is all about the energetics of protein folding. But I just want you to remember, this is like a reaction coordinate in 3 dimensions. So think about it being like this giant funnel that stuff can fall into, and they literally call this a folding funnel. And as you go down in the folding funnel, you go lower in energy. And we know that as we make intermolecular forces, we get lower in energy. And the idea is the folded functional protein is likely to be down at the bottom of one of these wells. What can happen is you can get trapped in one of these other spaces. And that might mean that you are folded, but not in a functional way. We didn't get a chance to talk about this. We'll probably revisit it. I'm trying to think if it's this semester or next semester, but talking about the plaque formation that happens with Alzheimer's disease. What happens is you have these functional proteins that end up misfolding and causing filaments. So just like we learned about with hemoglobin, if you have a mutation, that can cause a formation of filaments because you have things that are interacting that normally wouldn't, and that's not favorable. Don't worry about collagen. Don't worry about these 3 slides because we didn't get to them. But at some point, maybe I'd love to have a conversation if anybody's interested in talking about, hey, if I was to take a collagen supplement, what do I need to think about? Why is gluten problematic for some people? And there's videos that are out there too. We just didn't have time to get to it. And then this concept of how Alzheimer's disease works and why there is a correlation between glucose dysregulation and the phenotypic development of Alzheimer's disease. So things that we didn't really get to. And then here's all the slides from the insulin case study. And I think, yeah, that's all the rest of the slides. So by and large, a lot of times with the case study, you don't need to know all the details from case study. Here's some of the things that you need to know. Let me go back to the beginning here. We will study insulin a lot, so I want you to be able to think about the structure of insulin. It is essentially a two-peptide protein held together with disulfide bonds, but when it is synthesized naturally, we actually have a C-peptide that acts as a folding guide sequence to ensure the correct formation of disulfide links. Okay. And we talked about the story of how you can use that C-peptide level to determine how much insulin somebody is making in their own body, particularly if they're a diabetic and they need to be taking insulin. It's the only way to tell whether they're making their own insulin. Some things that you should be able to apply to a new situation. If you are looking at amino acid mutations, be able to tell me whether it is for functional group changes that maybe, mutating a serine to a threonine is going to be a conservative mutation because you're still maintaining the same functional group, right? But changing from a glutamic acid to a lysine, that might be significantly impactful because you're changing from a negative residue to a positive residue. So be able to think about those things. I think one of your quiz questions was asking you with the 1-letter code, if I were to have these 3 amino acids, which would represent the most conservative substitution? And you had to look and say, all right, if I'm going to replace this amino acid with something else, what is going to be the best substitution that's going to keep maybe this functional group? So that's one of the things I want you to be able to think about. Some of these things are just applications of concepts we've already talked about, like PI values. We will be thinking about, we've done a lot in lab already with gel electrophoresis. So being able to think about gel electrophoresis, particularly thinking about 2 reagents that we've talked about. Beta mercaptoethanol is an unknotting agent, okay? It's something that breaks apart, and I thought I had this written down here. It is something that breaks apart disulfide bonds, whereas urea or SDS is something that unfolds proteins. So be able to think about what different chemicals do to a protein, what kind of structure they might be interrupting. Let's see. Oh, here. I knew I had it someplace. So urea is unfolding. Beta mercaptoethanol is unknotting. So I do want you to know those reagents and what they do. Here's that story about the C-peptide. If you guys haven't figured this out, I like to teach this class with stories because hopefully stories are easy to remember than just like random facts. Okay. So as you go through this case study, with some of the details, if it was largely told as a story, so see if that's a way that you can remember it. Anything else from those notes? I want to try to go through this last stuff fairly quickly because it's more recent. Let me see if there's anything on the homework. You should be able to draw a generic amino acid, right? Here's an example of drawing a peptide. Again, a lot of times with the homework, it's a way for you to synthesize or look at information from the notes in a different way, maybe sort of like a meta-analysis of things. So this is basically taking the amino acids that have pKa values and sort of thinking about them. Now, the only ones that we really didn't have or we don't talk about per se is even though thiol and phenol, the phenol group in tyrosine here have ionizable side chains in terms of their pKa value, we really don't think about them as such. Here, Maria, again, is nature conserves structure rather than sequence, the sterics and energetics pieces, contrasting the hierarchy of protein structure, alpha helices and beta strands. You know, you might see questions here where you're asking, where would a residue like to be? Thinking about the kinds of mutations and whether or not it would have a significant effect or not. This is comparing and contrasting NMR and X-ray crystallography. And then this is, again, the stability and specificity. So we've talked about a lot of these things, but there's just, you know, homework key that's giving you those answers. I'm going to try to go through this one fairly quickly because we've talked about this most recently. Here's our ligand sandwich pieces. So be able to think about those ligand sandwiches. We talked about apo versus holo. Oxygenation versus oxidation. I will say unit 4 more than any of the other ones in the past probably have the most extensive collection of videos if you needed to go back and sort of get a refresher on something. I'll be transparent with you guys. I use these videos when I need to refresh myself about the amazing things I want to tell you about. So I will watch them myself before I have class because that reminds me of all the things that I want to tell you that are important. So I'd encourage you to think about ones if they're particularly if you look at your notes and you're like, I don't know if I remember that. Bucket brigade. I'm not going to ask you a question about a bucket brigade, but it helped us think about how we role played, how oxygen delivery and carbon dioxide waste elimination sort of take place. There was a question that sort of asked you to think about ranges for things. Ranges for KD values basically are from 0 to affinity. KD represents the concentration of ligand you need to be at to be half bound with your ligand. So the smaller the number is, the tighter something binds. And so the limits of that would run from like infinitely small, 0, to infinitely large. Fractional saturation, all fractions run between 0 and 1. Big picture, be able to think about the difference between these two graphs that we saw. Hyperbolic binding for non-cooperativity, that's myoglobin. Halfway values are important. We've seen lots of halfway values so far, so that's what we are summarizing on this table. We are learning about binding of a ligand, and in this case with an exemplar or an example of myoglobin and hemoglobin. But we're going to see and translate a lot of the terms and things that we've talked about here to other systems. Now, what's going to be different with other systems is we might bind something that we then do chemistry on. That's what makes this a simple system to learn, is we're binding something that we don't do chemistry on. But the same fundamental principle is T state, not in a shape to bind your ligand. R state, we are in a conformation to bind your ligand. Again, just contrasting the structure between hemoglobin and myoglobin. And we starred these slides. Super important slide. Super important slide. There are videos, and I think they're only each about 10 minutes each if you need to refresh your memory on these. These are the 2 slides that you're going to want to use and speak to as you create your Flipgrid video. Again, that's going to be a good opportunity for you to review this, see how well you understand this process as if you had to create a deliverable that explains it. And it's all about these two slides. So I'm not going to say too much about those because they're complicated. They're involved. I encourage you to rewatch those videos if you need some a refresher on that. And then we role played this. Comparing and contrasting hyperbolic curves with myoglobin, which is non-competitive binding. And then we had sigmoidal curves, which represent cooperative binding, which is what we see in hemoglobin. And big picture thing is thinking about how we affect loading and unloading, which is this A region, and then transfer of oxygen between myoglobin and hemoglobin. Now we do a lot with looking at horizontal and vertical lines. So be able to think about what those mean. This horizontal line represents 50% saturation. The further to the right that I have to go to intersect my curve, the less tightly it binds. Okay, so we can look at how P50 values will increase. The higher the P50 value, the higher the concentration of ligand I'd need to be at to be half bound. That tells us that hemoglobin does not bind oxygen as tightly as myoglobin does, because myoglobin's curve is further to the left. Okay. And then the last things we sort of talked about were how do we shift these curves? So if this is sort of what the normal thing looks like for myoglobin and hemoglobin, how do we change these curves, right? We can change them with pH. We learned here we can change them by binding small molecules. We can change them by sort of modulating the functional groups that we see here, such that we are ultimately going to change the conformation. All of these decrease pH, stabilize with the T state with 2,3-BPG, and then adding CO2 to the N terminus. All three of these are going to stabilize the T state, which unloads more oxygen. Okay, that's going to be consistent with, hey, if we're doing a lot of activity, metabolic acidosis, pH goes down. Okay. If I'm doing a lot of metabolic activity, my fuel equation tells me I'm going to make a lot of CO2. This process, making these carbamates, is likely to be happening more frequently. So another way to favor the T state, dump more oxygen, which all of our, physiology is telling us we need to do. Then we talked about, modulating this by changing the environment. So what happens when we're at high altitude? So important is thinking about what happens under the normal situation, when we go to the top of the mountain, when we've lived at the top of the mountain, and then we return back to sort of normal altitude. So this was a lot yesterday. I know it was just yesterday, but give yourself an opportunity to see how well it stuck, and then you can rewatch a video if you need to. And then the last thing we learned about was sickle cell anemia. So that was really quick, sort of going through that. Let me take a quick glance at the homework and make sure that there wasn't anything in the homework that we didn't cover in the lecture or in their little study guide there. So again, talking about oxygenation versus oxidation, looking at hyperbolic binding curves. Thinking about the transit. So this is really when we did our role playing, when we started at the front of the room and then sort of went back to the back of the room. That was going from, heart and lungs to the tissues. And then we had sort of our return. So this was asking some questions about that. We've got sigmoidal binding for hemoglobin. And this is asking us about the position of all of these different points. And then here again is that 5 equation concept map, which you already had a slide on. So that's all of the homework that we have for Unit 4. And I'm already sort of past time from what I wanted to do. So let me just see if you guys have any other questions here. And then I'm going to show you the last resource that I haven't yet made available to you, but will be an important resource for you to utilize. Questions? Okay, here's the one piece that I'm going to give to you. And this, I started doing this several years ago and it works really well. Maria, you had asked what does the exam look like? How similar is it to the previous exam? And I said it's probably a 60-40 mix of what I would call like one-word answers. And the directions here tell you most answers require a 1 to 2 word answer, not a paragraph. And I literally give you a 1-inch line to write your one-word answer in, okay? Maybe it's circling one of the options. There are some problems that might ask you to explain or define or draw a structure. Answer in 3 sentences or less, okay? So, for example, this first question, and there's 7 points, which probably tells you there's 7 blanks that you need to fill in with answering things. Complete the following that relate to structure formation in DNA or RNA. I'm going to tell you what the first question is. The first question is something to the tune of, what is the name of the building blocks used to make DNA or RNA? Okay, so another question might be, what is the name of the linkage that connects the monomers that you had in the previous question? Okay, so those are the types of 1 to 2 word answers that you're going to have. I'm going to give you a strategy too as you're going through and doing your test. It's either going to be something that you'll know right away and you'll write it in and you'll move on. I'm going to encourage you, if you can't think of it right away, maybe circle it to make sure that you go back to it, but move on. Okay, if it's not something that you've got quickly from that instant recall, come back to it later. Okay, but there's about 60% of the questions that are like that. You can see here there's 5 points worth of questions on SNPs. Probably one of those things could be Dr. Perenni's going to ask me to define what SNP stands for. Single nucleotide polymorphism. Okay. In general, when I ask you for amino acid information, you have to provide me with the 1-letter code. For full credit, you have to provide me the 1-letter code. There's some things, proteins by the numbers. Some numbers like 40 or 4,000 might be relevant here. Maybe it's a number that you have to calculate either using a factorial or an exponent type of a thing. But most of these that you are seeing right now are going to be single word answers. Okay, so to that end, there's not a lot of writing for 60% of your test. It's recall of information. You can see that there's sections on fibrous proteins, protein function. Complete the following that relate to the function of globin proteins, ligand binding, relate to the functioning of your cardiovascular system. Okay, so that might be something to do with arteries and veins and, how things get circulated in your bloodstream. Okay, here starts some of the ones, and you can see there's only 13 questions that are there. Draw a tripeptide. I've got a cute little word that you have to draw. Draw this nucleic acid. Again, where my X's are, you're going to have to indicate, is it DNA or RNA? Are there 5' or 3' phosphates? Okay, and it asks you to label important pieces. And I just have this in quotes. The title of the question isn't Perot's model equilibrium equation slide, but I'm telling you, it's something related to that. I would know that slide, right? That is these slides that we starred. That one, and importantly, this one. Know those slides. And there is a reaction mechanism, one you haven't seen, okay? But I want to know, can you apply knowledge? Now, look at this. It's only 5 points out of the total of 100 points on the exam, so don't stress on it if you are still trying to master your mechanisms. But by the time we get to learning unit 8 and we understand enzyme catalysis, you want to make sure that you're shored up on mechanism by then, because that's where we then understand how chemistry happens in enzyme active sites by looking at reaction mechanisms. So we want to make sure that you're pretty good on it by that. So I will put this once I get the link from Zoom on the this recording. I will post that with this document in Teams so you guys have that. Okay? Questions? Sorry, I went a little bit over. Go ahead, Madison, you had a question.
Just for the protein by numbers, you said factorial was for when there's no repeats.
Correct. And a
Okay.
way to think about that, if you ever want to think, like if you forget how it works, just like run the story in your head. It's like, okay, I'm gonna have, you know, if I'm making a 100 mer, I'm gonna have 100 slots. If I have to figure out what am I putting in the first slot, I have 20 different options. And then for the next slot, I'd have 20 different options. Now, if I asked you to make a 20 mer, a 20 residue nucleic acid, and you're going to use each amino acid only once. Once I use my first amino acid for the first spot, that means I only have 19 options for my second. Anytime you're trying to figure out what's the possibility, you're multiplying together the number of choices you have for position 1 times the number of choices for position 2 times position 3 times position 4. Now, if they're different numbers for each of those positions, they're generally going to go down by one number. That's where that factorial piece comes in. If I have the same number for the options at each position, well, then I'm multiplying the same number together, which is why we use the exponent piece. Make sense? Anything else? All right. Well, thanks for staying a little bit later. And then of course, as you're studying over break, don't hesitate. You can send a question. We'll see each other on Monday before your exam as well. But plan on, and you guys don't even need to let me know. You can either show up 9 o'clock or 11 o'clock to take your exam on Tuesday the 15th. All right.
Thank you.
All right. We'll see you guys tomorrow. Bye.
Oh, do I still send you the email reminding you about the correct answers
Yes.
for the quizzes?
Yep. And I don't think anything else came up. So just send me that email with that one thing because I already forgot. So
Okay.
yes, yeah, and I will make sure I'll take a look at that and see if I can get that done before tomorrow. And
Okay.
I'll also put that as a just a reminder when I send out this stuff on Teams that you should be able to go in and check your answers and see what the correct answers were so that you can study from that too. That's a good those questions are a good example. Now, it was written as a multiple choice, but you can imagine that a lot of those could be something where it was a one-word answer
Mm-hmm.
or the multiple choice. You might just see a question where it's not multiple choice. You just have to provide me with that answer.
Okay.
Okay?
Also, what will be the best way you would recommend to study for this exam, given that it's like 3 units compared to like the last exam?
I would say if I were studying for this exam, I would look over the homework,
Mhm.
make sure that that all makes sense. And I know there's a number of videos that are out there, but particularly if it's one of the videos that you felt like, or one of the slides that's correlated with the video. And let me just, let me share my screen real quick again. And I know Madison's gone, but this will be sort of available if anybody wants to continue to watch it. I go to this, and let's go to, let's say, protein function. Right? If you feel like, hey, you know what, I understood the bucket brigade piece, and you'll see a picture of what the slide is. If you're like, I got that slide, I don't really need any help on that, you're good. Yeah, I understand oxygenation and oxidation. I'm good. Oh, these graph ones, when we started doing that graph stuff, that's where I got a little bit lost. Okay.
Mhm.
You click on here, and you can be like, all right, I'm gonna just look at that slide again. This is Dr. Parenti's version of the slide. It's actually the same slide that I use in the video. And if you're like, I need some help, I'm gonna go here and I'm gonna watch this video which,
Customize and.
okay, well, this is not a bad thing. Ooh, look at it, 500
So what is ClickUp? ClickUp is
so
an all-in-one productivity software.
599
where you can manage
subscribers.
everything and
But-
anything.
you have to watch commercials now. So the key is, it's like, okay, if I want a refresher on what this slide was about, you know, most of these videos are on the, you know, somewhere between 8 and 12 minutes. So they're not overly cumbersome, but especially since you have a week here to study, maybe take, you know, 1 or 2 videos each day and rewatch them and make sure that that story is stuck up here.
Okay.
Because for the most part, if you had access to these videos, you could watch them as you're taking the test, all the answers would be there.
Okay.
And I would also look at the homework, right? So looking at the homework, you know, pretty much every answer or every test question that you have, I could look back and there's nothing that is on the test that is not in the homework, in my annotated notes, which also should reflect your notes, okay? Or those videos.
Okay.
Everything is there. So, and I know that's still a lot, but I would say if you're looking for a way to just make sure that you're shored up, make sure that you could redo the homework. If there's big picture concept slides that you're struggling a little bit with, rewatch them on YouTube. And then that should probably be good. And look at my annotated notes and make sure that there's not anything missing from your notes.
Okay.
Helpful?
Mhm.
All right. I'll see you tomorrow.
Okay. See ya.